Usually, we think of the egg cell as this, ultimate blueprint of life. Like it's a lone, heroic cell that just inherently knows what to do.
Right. It has the DNA, it has the instructions.
Exactly. And it supposedly just executes the whole plan entirely on its own.
It is, it's a very romantic idea, right? The solitary journey of a highly programmed cell. But if you actually zoom in on a fetal ovary, that lone hero narrative just completely falls apart.
It really does.
Yeah, it turns out that the survival of our species actually depends heavily on, well, cellular office gossip.
And that biological gossip is exactly what we are looking at today. We are taking a deep dive into the very beginning of female meiosis, which is the microscopic cellular process that ultimately creates egg cells.
The origin story of the egg, essentially.
Right. And our guiding source for this is a fascinating 2025 research paper from the journal Cell, Death, and Disease. It's titled, Somatic Kitl Promotes mTOR to Facilitate Prophase I of Meiosis in Female Embryonic Gonads.
Which sounds incredibly dense, I know.
It is a mouthful, but we are going to unpack it completely. We're looking at how the surrounding nurse cells in the ovary actively guide the main germ cells into this massive genetic event. And this new research, it actually overturns a textbook model of developmental biology that we have relied on for years.
It completely shifts our understanding of who is actually in charge of reproduction at a cellular level. But to appreciate the magnitude of this shift, we really first need to look at the stakes of meiosis itself.
Because it's not just regular cell division.
No, not at all. Most cells in your body divide simply by copying everything. You start with 2 sets of chromosomes, you end up with 2 sets. But a germ cell, the precursor to an egg or a sperm, it has to do something incredibly dangerous.
It has to cut that genetic material exactly in half.
Precisely, because when an egg and a sperm eventually meet, they each have to in 1/2 to create a complete set for a new human being.
Right, which makes sense mathematically, but biologically, that halving process is, it's fraught with peril.
Oh, absolutely. If the chromosomes don't pair up and separate perfectly, the resulting cell will have the wrong number of chromosomes, or damaged DNA. And that leads directly to infertility or miscarriage or really severe congenital anomalies.
It's a high-stakes genetic reshuffling.
Exactly. And in female mammals, like the mouse embryos used in this specific study, this dangerous process kicks off very early in fetal development, specifically between embryonic days 12.5 and 14.5.
Which is a remarkably tight window. And, that brings us to the textbook myth we mentioned. Because for the longest time, biology textbooks told this very simple story about how this process starts.
They really did. They claimed that a chemical called retinoic acid, or RA, was the sole ultimate trigger for female meiosis.
Right. The old model said that this wave of retinoic acid just washes over the fetal ovaries and turns on a critical gatekeeper gene called STRAY8.
Yeah, and STRAY literally stands for stimulated by retinoic acid 8. That name alone tells you just how entrenched this idea was in the scientific community.
Because biologists knew that without STRAY8, meiosis it simply doesn't happen.
Right, the cells just sit there in biological limbo. So the assumption was very logical. If STRAY8 is the gatekeeper and retinoic acid stimulates STRAY8 in a petri dish, then RA must be the spark that starts the fire in the living body.
But wait, if that's what the textbooks say, why is this new paper challenging it? Like, there must be a crack in that old foundation somewhere.
The crack is actually a gaping hole, to be honest. Recent studies began looking at what happens in a living, breathing organism, what we call in vivo, rather than just looking at cells isolated in a dish.
But what did they find when they looked in vivo?
Well, researchers genetically engineered mice to lack all the receptors for retinoic acid. So, in theory, without the ability to receive that RA signal, meiosis should have completely failed.
But it didn't.
No, instead, those female mice initiated meiosis perfectly fine.
Wow. So retinoic acid is essentially like a starter pistol that works perfectly in a laboratory setting. But in the real world, the runners are starting the race even when that specific pistol isn't fired.
That's a perfect analogy. There has to be another signal, a completely different hand, turning the key to open that STRAY8 gate.
Because the germ cells aren't just waiting for a generic chemical wash like RAA.
Exactly. They are actively prompted by their immediate physical environment. They're actually listening to the specific neighboring cells they are touching.
Okay, let's pause here for a quick recap. So the old rule that retinoic acid triggers meiosis is basically flawed when we look at living organisms. The germ cells need a different external signal to open the STRAY8 gate. And for you listening, it really makes you wonder, right? Have you ever discovered that a strict scientific fact you learned in school was actually just a simplified version of a much wilder truth?
It happens more often than you'd think in biology.
So since retinoic acid clearly isn't the whole story, how did the researchers find the real trigger?
Well, they decided to literally read the chemical text messages these cells were sending to each other, and they used a technique called single-cell RNA sequencing.
Which is a massive leap in molecular biology, right?
Yeah, huge. Previously, analyzing a piece of tissue was a bulk process. You would take the whole fetal ovary, mash it up, and look at the genetic material all together.
Right, I always think of bulk sequencing like putting a whole fruit smoothie into a machine and trying to figure out what individual fruits went into it. You only get the average flavor of the whole blend.
Yes, exactly. You lose all the nuance. But single-cell sequencing lets you look at the individual strawberries, the bananas, the blueberries before they ever get blended.
By isolating the fetal ovary at that critical day, 12.5 to 14.5 window, and separating every single cell.
Right. And then researchers can read exactly which genes are turned on or off in a specific cell at that exact microscopic moment. They're essentially mapping out the cellular communication network.
And when they did this, they identified two very distinct populations of cells interacting, right?
They did. First, they found the main characters, the germ cells, which are the precursors to the actual eggs. And the researchers could easily spot these because they uniquely express a marker gene called VASA.
Okay, VASA marks the germ cells. What was the second group?
This second group is the supporting cast. These are somatic cells, meaning non-reproductive cells, called granulosa cells. And these are marked by a gene called FOX2.
FOXL2. So granulosa cells are essentially the nurse cells.
Exactly. They structurally surround the germ cells, basically creating a physical niche for them.
So by using specialized software to analyze the signals flowing between the granulosa nurse cells and the germ cells, what did they actually find?
The discovery was loud and clear. The nurse cells were acting as senders. They were actively pumping out a very specific chemical signal called Kitl, that's K-I-I-T-L, or KIT ligand.
And what were the germ cells doing?
Meanwhile, the germ cells were acting as receivers. They were expressing the receptor for that specific signal, which is known simply as KIT.
It operates as a highly specific lock and key mechanism then.
Precisely. The granulosa cells hold the key, which is Kitl, and the germ cells have the lock on their surface, which is the KIT receptor.
I'm still trying to wrap my head around the implications of this. I mean, the germ cells are the VIPs, right? They're the ones carrying the entire genetic future of the species.
Right.
Yet they aren't actually calling the shots. They're just sitting there waiting for permission from the surrounding support staff to begin their most important job.
They absolutely are waiting for permission. And from an evolutionary standpoint, it is actually a brilliant strategy.
Why is that?
It comes down to safety. Like we discussed, meiosis involves a cell intentionally breaking its own DNA to reshuffle it. You do not want a cell initiating that catastrophic process unless the environment is absolutely perfect.
Oh, I see. So the somatic granulosa cells act as, like, environmental sensors.
Exactly. They monitor the structural and chemical readiness of the entire ovary. And only when they deem the environment perfectly secure do they release Kitl.
So that Kitl signal basically tells the germ cells, the niche is secure, you are cleared to begin the genetic reshuffle.
You've got it. That's the exact message.
That sender-receiver dynamic is incredible. Let's do a quick recap of this part. The granulosa nurse cells use the Kitl signal to talk to the KIT receptor on the germ cells, giving them the green light for meiosis. For you listening, what stands out to you about how dependent our fundamental biology is on this kind of microscopic teamwork? We really don't operate in a vacuum, even at the level of a single cell.
We really don't. But, in science, just observing a signal flowing from a sender to a receiver isn't enough to prove it's the main driver.
Right, you have to prove it's actually doing the heavy lifting. So to prove Kitl is the true trigger, the researchers had to break the system.
Exactly. They needed to see what happens to meiosis when that specific signal is entirely taken away. And to achieve this, they created a conditional knockout mouse model.
Okay, wait, why conditional? Like, why not just engineer a mouse that doesn't have the Kitl gene at all?
Well, because Kitl isn't uniquely used in the ovary. It's a critical signaling molecule used in all sorts of other developmental processes like blood formation, even skin pigmentation.
Oh, so if you delete the Kitl gene from the entire mouse from day one,
the embryo likely wouldn't survive to the stage where you could even study its ovaries. So they had to use a genetic trick. They engineered the mice so that the Kitl gene was deleted only in those specific FOXL2 positive granulosa nurse cells.
Ah, I see. So the rest of the mouse remained totally normal. They essentially just snipped the phone line exclusively from the nurse cells.
Exactly. And when they checked the ovaries a few days later, the results were an absolute disaster for meiosis.
What happened?
Well, the first thing they noticed was that significantly fewer germ cells ever managed to turn on that street gatekeeper gene. The gate just stayed closed for a huge chunk of them.
But some of them did manage to get through.
Yes, but for the germ cells that did somehow try to proceed, things got incredibly destructive during a phase called Prophase I.
Prophase I is, that's the heart of the meiotic process, right?
It is. It's where homologous chromosomes, meaning the copy of chromosome 1 you got from your mom and the copy from your dad, they have to find each other, pair up tightly along their entire length, and physically swap pieces of DNA.
And that swapping process is called recombination.
Yeah.
It's the reason you don't look exactly like your siblings.
Exactly. Now, to manage this physical pairing, the cell builds a literal scaffold between the chromosomes, which is called the synaptonomal complex.
Okay, and there are two key structural proteins here, right? Syntp3, which forms the side rails along each chromosome, and Syntp1, which forms the rungs connecting them across the middle.
Right. The architecture is very, very precise. And without those structural proteins locking the chromosomes together, recombination just cannot happen safely.
It sounds exactly like a zipper on a jacket. Like the Syntp3 proteins are the fabric tape on either side, and the Syntp1 proteins are the tiny metal teeth pulling the two sides tightly together.
The zipper analogy works perfectly here. In these conditional knockout mice, without the Kitl signal arriving from the nurse cells, the researchers found that the zipper basically failed to form.
Because the cell couldn't produce enough 6p1 and 6p3.
Exactly. So the zipper was missing its teeth and it got stuck halfway. The chromosomes couldn't pair up tightly and the cells became physically trapped in this intermediate stage, unable to progress. The whole genetic jacket was ruined.
And I mean, a broken zipper is bad enough, but the consequences are actually much more severe, aren't they? Because of how that DNA swapping mechanism works.
Yes, it gets much worse. Because to trade genetic material, the cell actually has to intentionally slice its own chromosomes, creating double-strand breaks.
Intentionally fracturing your own genome sounds like a terrifying biological event.
It really is. To manage this safely, a healthy cell relies on a highly coordinated emergency response system. Normally, specific proteins act like flashing red sirens.
Like the protein RAD51, right?
Exactly. RAD51 rushes to the site of the intentional break to mark the damage. And almost immediately, specialized repair crews arrive at those sirens.
These are proteins like DMC1 and MLH1.
Yes. Their job is to bridge the broken DNA strand to the opposite chromosome, facilitating a safe swap and then sealing the break.
But when the researchers looked at the germ cells that were deprived of the Kitl signal, it was pure chaos, wasn't it?
Complete chaos. They saw that the RAD51 sirens were blaring everywhere. The DNA had been sliced open all across the genome. But the repair crews,
The DMC1 and MLH1 proteins,
they were completely missing. Their numbers had just plummeted, so the double strand breaks were just left open and unrepaired.
wow. So the cells were essentially bleeding out genetically.
They really were. And here's the crucial biological insight from all that devastation. When a cell recognizes a massive amount of broken, unpaired DNA, it doesn't just keep blindly trying to divide.
It triggers a fail-safe mechanism, doesn't it?
A profound failsafe. It halts the entire meiotic process. The cell essentially makes a systemic decision. We cannot pass this fundamentally shattered DNA onto a potential embryo. And it arrests its own development.
Which is exactly why the loss of that external Kitl signal is so catastrophic.
Exactly.
Okay, time for another quick recap. Removing the Kitl signal completely ruins the chromosome zipper, those SYNCP1 and SYNCP3 proteins, and it leaves the DNA shattered without its repair crews, triggering a massive failsafe. So for you listening, if you were engineering the human body, wouldn't you want a strict, unforgiving failsafe exactly like this to stop severely damaged DNA from ever becoming a fertilized egg?
It is a brilliant, necessary defense
It
mechanism.
really is. So, okay, we've established the external signal, the Kitl knock on the door from the nurse cells, and we know the result, the fail-safe lockdown, when the zipper and repair crews fail. But I'm losing the thread a little bit on the middle part.
The internal wiring.
Yeah. How does an external knock on the outside of a cell physically build a chromosome zipper on the inside?
That is the black box the researchers had to illuminate next. They had to trace the internal wiring of the germ cell to really understand the signaling cascade. Because when Kitl binds to the KIT receptor on the cell surface, it doesn't just, magically teleport instructions to the chromosomes in the nucleus.
It has to trigger some kind of chain reaction.
Exactly. A highly specific mechanical chain reaction inside the cytoplasm. The KIT receptor immediately activates a protein called AKT.
Okay, so AKT is the first domino falling inside the cell.
Yes. And once AKT becomes active, it reaches out and turns on a massive, famous biological pathway known as the mTOR pathway.
mTOR. In cellular biology, mTOR is widely recognized as a master regulator of cell growth and metabolism, right?
It is. You can think of the mTOR pathway as a high-powered manufacturing factory inside the cell. When mTOR is switched on, it drastically ramps up the translation and production of proteins.
Let me see if I can synthesize this. The factory gets turned on and it starts churning out the exact proteins we just talked about.
Exactly.
It starts manufacturing striate, the gatekeeper. It churns out 6p1 and 6p3, the zipper teeth, and it pumps out the Vasa marker. It's basically a biological Rube Goldberg machine.
That captures the mechanics beautifully. The Kitl signal is the key turning the lock on the surface. That lock revs the AKT engine, and the engine powers the mTOR factory to physically build the zipper pieces.
Okay, but how do they prove that this internal factory was actually responsible?
Well, the researchers designed two brilliant experiments using fetal ovaries cultured in a lab setting. First, they applied a drug called rapamycin.
Ooh, rapamycin. That's pretty well known as an immunosuppressant used in organ transplants, isn't it?
It is, yes. But at the cellular level, rapamycin does something very specific. It is a potent inhibitor of mTOR. It essentially shuts the cellular factory down completely.
So what happened when they used it?
When the researchers applied rapamycin to normal, perfectly healthy ovaries that had plenty of the Kitl signal knocking on the door, it caused the exact same meiotic failures as removing Kitl did.
Wow.
Yeah, the zipper proteins disappeared, the DNA breaks piled up, and the cells hit the failsafe.
So even if the nurse cells are frantically knocking on the door, if the factory inside is broken, the whole process just grinds to a halt. The outside signal doesn't matter at all without the internal machinery.
Exactly. But the second experiment was even more profound. It was basically a rescue mission.
A rescue mission.
Yeah. They took the genetically modified knockout mice, the ones missing the Kitl signal where meiosis was totally failing, and they exposed those specific cells to a chemical called 3-BDO.
And what does 3-BDO do?
It is a direct mTOR activator. It forces the factory to turn on, completely bypassing the need for the AKT engine or the Kitl key on the outside.
And let me guess, it worked.
It worked beautifully. By manually turning on the mTOR factory, they rescued the dying cells. The protein levels of the striate gatekeeper and the synapse zippers spiked right back up, and the cells successfully restarted meiosis.
That is unbelievable. It proves exactly how step-by-step and almost industrial this entire biological process really is.
And when you step back and look at this entire mechanism, this Kitl-Kit-mTOR axis, it reveals something really profound about how biology operates.
Which is?
It proves that this entire system operates in parallel with the old retinoic acid model.
So biology rarely relies on a single point of failure for something as critical as reproduction.
Exactly. Instead, it builds robust, multi-layered regulatory systems. The retinoic acid pathway and the Kitl pathway, they basically operate like two separate verification systems working in tandem.
Like turning two keys at the same time to launch a missile.
That's it. They ensure the cell only enters the dangerous process of meiosis when conditions are absolutely undeniably optimal.
Okay, quick recap of the factory. The Kitl signal activates AKT, which turns on the mTOR factory, which builds the meiotic proteins. And we know this because blocking mTOR ruins meiosis, while artificially activating it saves it. So for you, the listener, does knowing how mechanical and step-by-step this process is change how you view the so-called magic of reproduction? It replaces that mystery with these rigid, awe-inspiring mechanics.
It really is awe-inspiring. And, this brings us to the ultimate test in biology.
Right, because if scientists deeply understand a mechanism, they shouldn't just be able to break it or rescue it in a living ovary. They should be able to build it entirely in a petri dish.
That is the gold standard. If you truly know the rules of the biological game, you should be able to play it outside the body. So the researchers took primordial germ cells, these are raw, uncommitted precursor cells, and isolated them in a culture dish.
Now, if they just follow the old textbook recipe, giving those raw cells retinoic acid and another standard molecule called BMP2, what happened?
The results were incredibly poor. the cells attempted to enter meiosis and, a little bit of that stray gatekeeper protein was produced, but the process was highly inefficient.
The chromosomes probably failed to pair up correctly.
Yes, the zipper proteins were virtually non-existent and the cells were defective. It proved beyond a doubt that the textbook formula of RA and BMP2 is simply not enough to sustain the process.
But then, they added the missing ingredient.
They introduced Kitl to the culture, and the transformation was dramatic. Those isolated germ cells suddenly began to aggregate together beautifully, mimicking the solid tissue structure of a real ovary.
Oh, well.
There was a massive, statistically significant spike in normal chromosome pairing. The production of the stray gatekeeper proteins and the synstripid zipper proteins just went through the roof.
So they successfully orchestrated the origins of a healthy egg cell entirely in a laboratory incubator, just by providing the correct sequence of chemical whispers.
Yes. And beyond just proving their theory, this opens enormous doors for the future of reproductive medicine.
Oh, I bet. Because if we know the exact chemical language required to safely mature a primordial germ cell into a functional meiotic cell, we can vastly improve in vitro models.
Exactly. This has massive implications for understanding the root causes of infertility, or conditions like premature ovarian failure, where the delicate signaling environment between nurse cells and germ cells might be disrupted.
It blurs the line between biology and engineering in the most incredible way. All right, let's tie all of these pieces together for our final recap. By adding Kitl to raw germ cells in a dish, researchers successfully recreated normal meiosis, proving its essential role and opening new doors for treating infertility.
It's a huge leap forward.
It is. And I want you, listening right now, to think about your own existence for a second. Every single person exists because decades ago, in your mother's fetal development, this exact, highly choreographed chemical dance happened flawlessly.
That's exactly right.
A microscopic nurse cell sensed the environment and released Kitl. It bound to a germ cell, activated the AKT engine, cranked up the mTOR factory, and zipped up the DNA perfectly.
And if any single one of those dominoes hadn't fallen exactly right,
the egg that became you would have hit that brutal cellular failsafe and stopped existing before it ever had a chance.
We are quite literally the product of billions of successful microscopic cellular conversations.
I want to leave you with a lingering thought to mull over. We always think of germ cells, the sperm and the egg, the physical carriers of our DNA, as the ultimate masters of our genetic destiny. They are the undeniable stars of the show.
Or the lone heroes.
But if these surrounding somatic nerve cells actually hold the keys to initiating life, if they're the ones sensing the environment, judging its safety, and ultimately giving permission for the DNA to shuffle, how much of what we consider inherently dictated by our genetics is actually controlled by our cellular environment? Who is really in charge?
The blueprint is only as good as the cellular construction crew reading it. It's a perfect question to ponder.
Until next time, keep diving deep.